Good day. In this lecture, we will be discussing about multiple sequence analysis. But first, we must identify what exactly is multiple sequence analysis. So multiple sequence analysis, or we can say it as alignment of multiple sequences. So this is a collection of 3 or more proteins or even nucleic acid sequences that are partially or completely aligned. When we say partially, because we align them based on local alignment, whereas if it's completely aligned, it's a global alignment. It's just like pairwise alignment, but instead of just having a pair or two sequences that we align together, in multiple sequence analysis, as the name suggests, we are aligning more than two sequences, so 3 or more. So this sequences, so why do we need to align 3 or more sequences? Most of the reasoning is they are either evolutionary or the sequences are related in an evolutionary sense. So that means they are presumed to be derived from a common ancestor as per the theory of evolution. And also, it can be can also align them based on their structural identities. So when we say structural identities or in the structural sense, these sequences, they can both be DNA, RNA, even nucleic acid. So the nucleic acids comprises not only the DNA but also the RNAs. Um, they have similar structures. So we can apply them on, especially on proteins. Protein, uh, Previously, the identification or the prediction of protein structures, three-dimensional structures, rely on multiple sequence analysis. So if we have sequences of proteins that have known um, motifs or known structures, we align another structure to that one with we align another sequence with a known structure if they have similar batches, or we have um, segments of their sequences that are very much aligned. Most likely that segment have the same structure. So that's the idea with a structural prediction. That's why we tend to align them. This is one of the most common, actually, one of the most common applications when we do sequence analysis. It's not only the pairwise. We seldom do pairwise analysis, but more often than not, in actual application, in actual research, we do multiple sequence analysis because there are a lot of information that we can get from multiple sequence analysis as compared to pairwise. Okay, so in the structural sense, so the aligned residues tend to occupy corresponding positions in the three-dimensional structure of the aligned protein. But take note, the structural analysis for multiple sequence and the structural sense for aligning multiple sequences is not only limited to protein. DNA also has their own three-dimensional structures. In fact, some of the regulatory elements in the DNA, when we say regulatory elements, these are regions in the DNA that do not encode for a specific protein. So they are not genes, they are non-gene coding regions, but they have a function to regulate the activity of genes, such that these are the regions where your transcription factors, your silencers, your operators, your what other enhancers, transducers, these regulatory elements bind. And in order to bind, especially in the field of biology, it's the structure that is very important. The structure of a molecule provides the basis for the function. So if you destroy the structure of the molecule, it loses its function. And that is why in this field of study, it's very important to have an idea of the structure of these molecules, these sequences, and so as we can say now what is its function. And hence, that's why we do this sequence analysis. So why do we do this sequence analysis, as I was saying before? So if you have a protein or even a gene that you are studying to is related to a larger group of proteins, so this group membership can often provide insight to the likely function. So if you have a protein or even a gene that you have no idea what the function is, and but you do know the sequence, you align it versus the database, and then you can have an idea, especially if it's a novel gene or of a novel sequence, a novel protein, if it is distinctly related to a larger group of proteins. You can now say that this one is a hexokinase, or this one is a type of a kinase. This one is a type of an enzyme catalyst. So let's say hydrolase. So this is actually how these proteins, these enzymes are often identified. So, because of course, in actual laboratory experiments, it's one of the challenges is to study the functions. And this is one of the shortcuts so that you don't have to do very extensive screening because you have no idea where to start. So you can start with, for example, let's give you a scenario. You have this novel bacteria that you analyze. You do proteomics analysis on that novel bacteria. You found several distinct novel proteins. You don't know what these proteins are, but you can sequence those proteins. That's easy to do, but you don't know what they do, what are they for. So you can run a blast search, or even align them on several groups of similar proteins, or not say similar, but families of proteins. And for example, you have this unknown A. You align them, and then it turns out that this is a type of they are very similar in sequence to the kinases. So that means there's a high probability that that protein is actually a kinase. So we will be the next step would be to verify. So instead of doing multiple tests because you have no idea exactly where to start doing the test, so you start with everything from hydrolases to kinases, which is actually very time consuming, not to mention resource consuming. You just focus on its activity as a kinase. So that is to verify whether that protein really is a kinase. So that's one reason why we do multiple sequence analysis. So aside from that, we can have this we can use them to also detect for homologs. So we have discussed homologs previously in our pairwise alignments lectures. So how basically this is just doing similarity searches, but instead of just two proteins, you get three or more proteins. So we get multiple profiles for them. And then we can also be, when the output of any database search, such as your BLAST search, is examined, a multiple sequence alignment format can be extremely useful. Actually, actual BLAST search, which you have done in some of your laboratory activities, if you did it up until the end, uh, you will see there that when you input a query sequence in a BLAST, and we do not identify it as a pairwise alignment, you will end up with several hits. Actually, not several, many hits. And if you look at their graphical analysis, or this alignment format output, you will see there that you have several red lines. That means these are the aligned sequences. So that is one of the output. The BLAST does not do um, an actual multiple sequence alignment, but it does a pairwise alignment on your query versus all of the contents of the database. And that is its output, which is quite similar to the multiple sequence alignment, but not exactly. But because when we say multiple sequence alignment, we are actually aligning uh, three or more sequences all against each other, instead of BLAST, which is actually doing it as one versus all, instead of all versus all, which is that is the true sequence alignment. Multiple sequence analysis is actually all versus all type of alignment, versus the BLAST, which is one versus all. So basically, that is the difference. And also, some of other reasons to do multiple sequence alignment is that the human genome harbors 11,000 non-synonymous single nucleotide variants and counting, which they are caused by some amino acid substitution, some of which, around 300, are predicted to be deleterious. So we can have algorithms that predict whether the variants are harmful, or often rely on DNA and or protein multiple sequence alignment to assess cross-species conservation. So this is more of detecting diseases, genetic diseases. These are some of the applications of this one. And then deleterious variants that tend to occur at conserved positions. When we find variants, variations, or mutations in conserved positions, we can be alerted to some novel genetic diseases, or even some, it can be another adaptive behavior or adaptive mutations on that particular regions. Actually, you can see here, during this pandemic period, especially during the lockdowns, a lot of scientists have switched to studying the genome and the protein structures of the COVID, the SARS-CoV-2, and many of the variants have been shown to have mutations, especially some deleterious mutations on the spike protein. So some of them are deleterious. That means the spike protein changes in shape, and that makes it somehow evade some immune system parameters, and that is why we need to have updated vaccines and boosters, because the the virus is changing and we need to keep up with the changes. Okay, so aside from those things, we can have analysis of population data that can provide insights into biological function. In fact, there has been some controversies in that regarding even vaccine technology, because sometimes, because of course, uh, in other countries or in society, we can say this is a racist remark, but in reality, racism is an issue, especially with biology. Because, for example, this vaccine is effective only on Caucasian people, but not as effective on Asian or even Hispanic or even African um, people with African descent. And that is actually a true issue. That's a true and a real issue. So we need to have population data so that we can tailor vaccines and even therapeutic approaches to specific races, to specific genealogies of people, even specific populations. So you have that. And then, when the complete genome of any organism is sequenced, a major portion of the analysis consists in defining protein families to which all the gene products belong. So if you discover a new species, the next step, of course, is to analyze its genome. Then you have database searches effectively perform multiple sequence alignments, allowing comparison of each novel protein to the families of other known genes. Other than that, we have phylogeny algorithms that begins with multiple sequence alignment. Later on, we will talk about phylogeny. But at the start of every phylogenetic analysis, we do first multiple sequence alignments. We use the data to generate the trees, the phylogenetic trees. So the most critical part of making a tree is to produce the optimal multiple sequence alignment. And lastly, the regulatory regions of many genes. This is where what I was saying a while ago about the three-dimensional structure of the DNA and even the RNA. So the three-dimensional structure, not only the information encoded in the genes is important in the DNA. The DNA is also important not only because of the genes but also on the regulatory regions. And these regions are they have very conserved sequences. They have consensus. When we say consensus, they are very similar across the board. That means several species have the very similar consensus sequences. In fact, all eukaryotes share the same consensus sequences for the promoter. The same goes for the prokaryotes. You have the Tata box, the Taat box. You have those, the Pribno box. So those are regulatory regions where the promoter bind, and they are very highly conserved across not only species but across taxonomic kingdoms. So many such regions are identified based on the conserved non-coding sequences that are detected using multiple sequence alignment. So that is why multiple sequence alignment is actually quite important. And even though when you do, if you want to do thesis in or even research in bioinformatics, this is usually the first step when you do bioinformatic analysis. In the journals that you will be reporting on, you will see here that MSA is usually part of the methodology, but it does not, of course, be the only methodologies. Just part of the methodology of analyzing sequences. Okay, now let's look at how do we approach or how do we actually do multiple sequence analysis. So we have these five main approaches: the exact methods, we have progressive alignment. That this is the approach done by the program ClassTel W. This ClassTel W is the program, the MSc program that is housed or made by EBI, the European Biotechnology Institute. So they are using ClassTel W or even the standalone ClassTel Omega. Okay, so they do progressive alignment. And then iterative approaches, these are done by these programs Praline, Interalign, and Muscle. So you might have heard of muscle algorithm. So this is one of the more common algorithms used. So of course, these are standalone programs. Praline is also a standalone program, but usually used by Linux users. So I'm not sure if there are any Windows program here. Muscle is a Windows-based program. Now, consistency-based method like MAFT and ProbCons. MAFT also is, I think this is Mega software. Probcon is another software. So these are algorithms. Actually, Muscle is also algorithms. Pralines, these are algorithms, but they are also programs. So this, let's just say that, for example, um, it's why is the program is also an algorithm? Basically, they started off as algorithm, and of course the proponents who made these algorithms, who developed these algorithms, created GUI so that many people can have access to, and that is why we have this software. So they just tend to name the software after the program that they generate. However, there are some programs like Mega software. They do multiples, or they can you can use different algorithms on the using those programs. So they can switch to different, they offer several approaches or several algorithmic approaches. Okay, and lastly we have the structure based methods. So this one is not as common as the other four because there are some technical difficulties with that one, but as its name suggests, the structure-based methods included three-dimensional structure informations. And we have an example algorithm for that. We call it the Expresso algorithm. Actually, this is under the T-Coffee. The people who created T-Coffee, which is the program for multiple sequence alignment, um, it also created this structure-based method, Expresso. So it's one of the functions of the T-Coffee algorithm. Okay, so let's look at these 5 approaches one by one. So let's start with the exact approaches. In the exact approach, we are using the dynamic programming described by Needleman-Wunsch in 1970 for pairwise alignment. Actually, exact approach is an extension of the pairwise alignment. That is why it's also the first method to be proposed, but not exactly used. You will see later why not. But in the exact approaches, it's more of an extension of the pairwise alignment. But instead of doing it in a 2 by 2 matrix, basically in just a pair of sequences, you do it with 3 sequences and more. So as it is a pairwise alignment method, so they employ dynamic programming, but instead of a 2 by 2 matrix, as I said before, you are using multi-dimensional matrix. That means if you are aligning 3 sequences, you are going to use a 3-dimensional matrix. And then in if you have 4 sequences, will have 4-dimensional matrix, so on and so forth as you increase the number of alignments. So in the exact approaches, this is the most computationally intensive because your goal here is to maximize the sound alignment score of each pair of your sequences. So in standard dynamic programming, the branching order of the guide, because this one is using dynamic programming methods, therefore it also has its own disadvantages as the dynamic programming method, such that if you have a guide tree that is suboptimal, the scoring parameters may cause gaps to be misplaced based on your guide tree. If you remember, in the pairwise alignment, we are trying to find the optimal based on the matrix. we are trying to find the alignment, which path we will take from the bottommost right side going to the top left side, going by the one that with the highest score always, the square with the highest score. So this is done for multiple dimensions, but again, if you have a different branching order, because it's no longer a two-dimensional matrix, it's three or more dimensions. So if there are some issues with the branching order, the alignment might be suboptimal. And in addition to that, because you are generating multi-dimensional matrices, the amount of time and computational resources needed to do this multi-dimensional matrix calculations is very difficult. It's not actually feasible in time and space, especially at the time that the exact approach was first introduced or actually proposed. The computers at that time, it's around 1970s, 1980s. They of course they are not that computationally powerful enough to do the exact approach that was proposed. And that is why we have this next alignment approach, which is the progressive sequence alignment. So this is the most commonly used algorithm, and it was used by the EBI's Clustal W. So it was first proposed by Fitch and Yasunobu in 1975 and described by Hogeweg and Hesper in 1984, who applied it in the alignment of the 5S ribosomal RNA sequences. So if you remember, the 5S ribosomal RNA sequences are used to create or actually deduce the so-called tree of life. Although there are some controversies regarding the accuracy of the use of the 5S ribosomal, but the 5S ribosomal is basically the go-to sequence if you want to align all of the living organisms, because all living organisms contain ribosomes, and it is actually a good metric to compare everyone's uh, genealogy with everybody else, because all living organisms have ribosomes. So basically, that is the use of this Y5S ribosomes are quite important in bioinformatics, especially in evolutionary biology. Now, they call it a progressive sequence alignment, because instead of doing multidimensional matrices, this entails just doing pairwise sequence alignment scores between your sequences. It can be protein, it can be nucleic acids, that is DNA and RNA. So you just do pairwise alignment first, and then you start with the two closest sequences. So for example, if you have sequence A, B, C, D, and E, and you have the two closest sequences is apparently sequence A and B, you first start with you create an alignment with A and B, and then you add the one that is next closest to them. Let's say it's sequence E. So sequence E is the one that is next closest, so you add sequence E, and then you add the next with the next closest. Let's say it's sequence C, and then finally up until the last closest sequence. So this is the progressive sequence alignment. So you do not generate a whole multidimensional matrix. You just align them pairwise, and then choose the two closest, and then add them based on the which two closest. Now, the benefit of this approach is that it permits a very rapid alignment of hundreds or even thousands of sequences, because it's just a simple pairwise, and then compare the scores, and then add them progressively. That's why it's called a progressive alignment. But the limitation of that is the final alignment that you would have depends on the order in which the sequences are joined. What if, for example, A and B are very close, C and D are also very close? Now, because you start with adding A and B, because these are the two closest, and then sequence C is the next closest. But after sequence C, the next closest is sequence E, and finally sequence D. Now, if the closest if sequence C and sequence D are very closely aligned, but you add them not as a pair, or you add them in a different order, or as I said, A and B first, then sequence C, then sequence E before you add the sequence D. Now, if the two C and D are quite closely aligned, but it's not well aligned, but when you align sequence C to sequence A and B, it's no longer the optimal alignment that would happen when you align C with D. that is where it's no longer the optimal alignment. Now, and because of this dependence on the order in which the sequences are joined, it's not guaranteed that you would have the most accurate alignment results. So that is one of the problems or issues or disadvantages with the use of this progressive sequence alignment. That is why we have this iterative approach. So iterative approach seeks to answer or seeks to improve the progressive alignment approach, especially on its limitation, which is again the suboptimal solution because due to the order of adding the sequences. Now, in this method, you are basically doing multiple progressive alignments. So, in these methods create initial alignment and then you modify this alignment to try to improve it and then compare the scores. So, in such a way, you overcome therefore the limitation whereas you are limited, the alignment is can be suboptimal because the order is wrong. So, this one, you do many progressive, you iteratively do progressive alignments until you refine the final optimum alignment. So, you have here, for example, this one. So, you have your sequences, you align those sequences, and this is your initial alignment. Now, you partition this one, divide it into sub-alignments, and then regroup them. You would now generate a second alignment. Now, this second alignment that is slightly different from the first one, you compare it with the initial. So, if the score for the second alignment, the new one is higher than the initial one, you replace it. But if not, you don't replace it. So you do this multiple times until you have a convergent solution or the optimum score is no longer being replaced. It is now consistent or does not it can no longer be improved. So this is a refinement method used by MAFT, the MAFT program. So this is your iterative alignment. Now, aside from these three, we have the consistency-based approach. So this takes a different approach compared to the other three, which is the first three, which is mostly an improvement of the previous one. Now, the consistency-based approach is that for your sequences X, for example, you have three sequences X, Y, and Z. If residue X aligns, if a residue in the sequence X Xi aligns with a certain another residue with Z, and that residue also aligns with the third one, which is Y, then therefore X also aligns with Y. So basically, that's the idea. Similar to the mathematical principle of A is equal to B and B is equal to C, therefore A is equal to C. So you start, it's basically looking at specific residue that is that is aligned quite well with one another, and you put that as an anchor and try to align the rest. So consistency-based techniques, they score pairwise alignments in the context of information about multiple sequences. So they adjust the score of x of I to y of I based on the knowledge that z of k aligns to both x and I and y of I. So this approach is quite distinctive because it incorporates evidence from multiple sequences to guide the creation of your pairwise alignment. So basically, they are looking for, well, quote unquote, the anchor sequences. You align this particular residues that is consistent across the different sequences you are trying to align, and then you do pairwise alignment based on that anchor. And that is the consistency based approach. So an example of an algorithm that do that is ProbCons. And last but not the least, we have the structure-based methods. So the difference, as I said before, the difference of the structure-based methods with your consistency with the first four is that in the structure-based methods, you are also including information from tertiary structure. And why is that? That is because based on experimental data, if even if we have sequences that are not very similar to one another, but if we look at their tertiary structure, they're very similar. An example is the human beta globin and the myoglobin. If you remember, in the previous activities, beta globin and myoglobin are not that closely related. I think it's just around 25 to 40% sequence identity. But if you look at their 3D structure, they are very much the same. The way the structure folds is very similar. And so we can say that they are actually related because in the first place, they do have the same function. They bind to oxygen through the heme groups. And from that, there's homology in that function, that they have homologous function. Therefore, they are related. But if you look at their sequence only, in a certain vacuum, if you look at the sequence, you cannot say that they are actually related because they have very limited sequence similarity. But if you look at the structure, there is where the similarity and the homology emerges. And that is where the structure-based methods come in. Now, because we cannot really take into account the sequences, the information from a sequence only. So the sequences information is cannot be taken in a vacuum. So we also need to consider other things. And one of those things is again the structure. So in the structure-based methods is that you also incorporate the information of the structure. You align them structurally, not only as a sequence. And of course, it's a very different type of approach now. And we have several advanced or a different set of algorithms to do that. So example of these algorithms is the praline, and then the module of the coffee, we call this one espresso. In RCSB, you can also see it, FATCAT. There are actually new structural alignment methods that are emerging right now. In fact, these methods, um, there might rather right now there might be some new methods that are being developed as we speak. So as we continue on through the years, we are trying to improve more on these methods. So these structure-based methods, since it comes from they are aligning different, not the sequence itself but the structure. The information can be used as a checkpoint or a recovery check or a QC check for your sequence alignment. So if you do a sequence alignment and then you do a structure-based alignment, then compare the two. Are they close? Did they have the similar results? So they can be used to assess how accurate your sequence alignment is. Okay, so that's the structure-based methods, and that ends the multiple sequence alignment lecture. So in the next lecture, we'll talk about molecular phylogeny.